we're gonna start we're gonna start chapter 7. When we were doing chapter 5, we learned and chapter 6, we learned about the fact that the cell membrane is made up of something called as phospholipid. It's a phospholipid bilayer. This membrane is selectively permeable. What does it mean to be selectively permeable? A good way to think about it is like a sieve, like a flour sifter. It allows certain things to go in. It allows things to go in and out based on, let me turn on the pen. It can allow things to go in based on size, polarity, something that's nonpolar versus polar. So let's look at the basics of this, and then we're going to look at how is it that food, nutrients, the macromolecules that you require, and all the different things that need to go into the cell to allow the cell to function. What are the different mechanisms for things to go into the cell, and what are the ways in which things are sent out of the cell? We looked at one of the ways things are sent out of the cell, which is an exocytosis process. The process is called exocytosis. Exocytosis. We looked at this in terms of when we were talking about how proteins are made and they get into a vesicle, and then the vesicle transports that outside the cell. We're going to look more in detail about all of this stuff in this chapter. So basically the phospholipids, if you remember from chapter 5, you have a hydrophilic head. These are the hydrophilic heads. Hydrophilic heads, and this is made up of that glycerol and that phosphate portion of it, and then it has two hydrophobic tails, and on the internal end, you're going to see the same thing. The tails kind of are in the middle and they form like a pack, and the heads are on the outside and the inside. These molecules are called amphipathic, which means it has both a hydrophilic as well as a hydrophobic region. The phospholipid bilayer is also called a fluid mosaic model. It is not entirely solid. It's not a solid fat. If you remember, we talked about the fact that since they are unsaturated fats, the two tails that are there, they're unsaturated fats. And because they're unsaturated fats, they tend to have a kink in them, which makes sure that they don't pack onto each other. And at room temperature, they're kind of a viscous liquid. There is also proteins embedded within the system, within the phospholipid bilayer, hence making it a mosaic. This is a good representation of what it looks like. Remember, our body has a ton of cells are usually in an aquatic environment. Our body has a lot of water-based material on the extracellular material. So the outside of the cell has to be hydrophilic, and the inside, this is the hydrophilic part, which is the phosphate head, and this is the hydrophobic part, which is the hydrocarbon tails. In order to allow for transportation to happen, there are a ton of proteins, channel proteins, that are embedded into the phospholipid bilayer. The basic rule is like dissolves like. Something that is polar will dissolve other things that is polar. Something that's nonpolar will dissolve other things that's nonpolar. If you mix them up, it's like mixing oil and water. So in order for the protein to be embedded in them, this layer of it, the layer that interacts, is going to be hydrophobic, the one that's interacting there is going to be hydrophobic, and the inside part is going to be hydrophilic, so that it can allow for hydrophilic material to go in and out. So all of this area is going to be hydrophilic. All of this area is going to be hydrophilic. Hydrophobic regions will be next to the hydrophobic regions, and hydrophilic regions will be next to the hydrophilic regions. If you freeze fracture the cells, so there are different types of proteins that are found within a plasma membrane. You are going to find internal membranes that are internal proteins that are on the inner side of the protein, of the membrane. If we assume this is a cytoplasmic side, which means this is inside the cell, and this is the extracellular cell, this part, which means it's outside the cell. Certain proteins will be only on the inside. They kind of stick outwards towards the inside. Some of them stick into the extracellular proteins. So there's internal proteins, there's external proteins, or there's integral proteins, also called as transmembrane proteins. Transmembrane meaning going through and through. If you were to freeze fracture them, that is actually, think of it like a zipper, and if you were to unzip it, proteins that are on the inside will just go towards the inside, and the proteins that are on the outside will just go towards the outside. The phospholipid, as the temperature cools down, the membranes would switch from a fluid state to a solid state. But this is not happening constantly in your membranes, and your membranes are not solidifying at different temperatures. The reason for that is because your membranes are very rich in unsaturated fatty acids. So your because they're unsaturated, the chain tends to form like a kink. So because it's forming that kind of a kink, the next one that's down here is not able to actually pack in. So are not able, and then the next one that's here is also not able to pack in. Like, let's say if this was saturated, it still cannot pack in because of that kink being formed. So because there is a certain amount of movement, and there is rollover movement constantly happening, that is what basically a lateral shift is because there is movement. It is not solidifying. Your membranes would not work very well if they just solidified as soon as the temperature changed. They're usually, a good way to think about it is like if you actually took like something like olive oil or salads and put it in the refrigerator, they kind of get this viscous liquid kind of texture, consistency, and that's what it is. That's because of the existence of double bonds. At least one double bond makes it unsaturated. So this is a good example for that, where you see because of the kinks, there's all this gap and everything is not packing in. Hence, the things are not packing in. They remain more fluid. Between these phospholipids, you're going to see a bunch of lipid rafts made out of cholesterol. The function of cholesterol here is that at warmer temperatures, the cholesterol basically restrains the movement. It does not allow for just every part of it to laterally move back and forth. And at cooler temperatures, it maintains a certain amount of fluidity, so it doesn't, like, everything doesn't tightly pack in. So it kind of acts like a little barrier in the middle, so it stops them from completely packing in at cooler temperatures, and at warmer temperatures, it stops them from moving apart as fast as possible, because as temperature increases, the speed of molecules also increases. This is what it acts like. In the body, we have homeostasis. So our internal temperature by various mechanisms is constantly controlled at 37.6 degrees Celsius. It is controlled at that temperature so that your phospholipid layer, your cholesterol layers are functioning at the optimal rate at which they're supposed to function. These are the lipid rafts made out of cholesterol. So basically, because I have cholesterol here, these two guys can move back and forth, but it is going to prevent these guys from moving back and forth. At the same time, when it's cooler, when it kind of stops this guy from compacting onto this guy and packing on. So it's serving both purposes. It's making sure that this guy doesn't like constantly move this way and this one is moving that way in warmer temperatures. And in cooler temperatures, it's trying to make sure that this guy doesn't pack onto this guy. If this is both of them are saturated, if both sides are saturated, it makes sure that they don't pack in on each other. This is a really good picture for you guys to understand what your plasma membrane looks like. Just to review, you have the hydrophobic heads. So this is the outside of the cell. This is the inside of the cell. You have just let me get the pen out. Okay. This is the outside of the cell, and this is the inside. Here are your hydrophilic heads, your hydrophobic tails, and then ever so often between them you have the cholesterol lipid rafts. Proteins that go through and through, like the ones here, are called integral proteins. They're also sometimes called transmembrane proteins. You have if the protein is only on the outside, then it's a peripheral protein, or the outside protein. Sometimes it's called external proteins, and sometimes it's called internal proteins, and these are peripheral proteins. These are either only on the outside or only on the inside. This is the glycolipids. The glycolipids act as receptors. They also act in a way that allows you to know what type of cells it is, for identification of the cells. Something like this is a glycoprotein. This is a protein, and it's and then there is a glucose molecule attached to it. Some of the glycoproteins that we looked at it in the previous chapter where we talked about fibronectin. Fibronectin is one of is from that category where they're always on the outside of the cell. And the external matrix, these fibers are collagen fibers and elastin fibers. Recaps everything. Peripheral proteins, they're bound to the surface. Internal ones are called internal peripheral proteins. External ones are external peripheral proteins. Integral proteins are the ones that they penetrate through the hydrophobic region. The part of the protein that is interacting with the hydrophobic region is the hydrophobic in nature. So they would have on the outside amino acids that are nonpolar, and the part that is on the top is going to be hydrophilic. So they would have polar amino acids on that end. The inside of these proteins are usually the inner part of these proteins. If they are channel proteins that are allowing for other polar molecules to go in, then the inside is going to be hydrophilic. So the best way for you to like realize this is like dissolves like. So if something is interacting with a polar molecule, then it is also going to be polar. If it is interacting with a non-polar molecule, then it is also going to be non-polar. Non-polar molecules are going to be hydrophobic, and polar molecules are going to be hydrophilic. This is a good representation, and from your chapter 5 you've realized on both ends you'll see they kind of corkscrew through the system, and on one end you'll have the N terminal, the amino terminal, where the amino acid is the amine group is connected. On the other end you have the C terminal, that is where the carboxylic side is connected. The membrane proteins basically are doing some of the most important functions that are required for the day-to-day functioning of the cell. So transport is usually done by transmembrane proteins. These are usually transmembrane proteins. I'm just going to call it GM. Enzymatics are usually done by, it happens on the inside of the cell. And so that is going to be done by peripheral proteins, peripheral internal proteins. Signal transduction is happening on peripheral, on the internal. Cell-to-cell recognition is going to be peripheral, external. Cell joining, the connections between cells is going to happen peripheral, external. And then you have attachment to the cytoskeleton. Cytoskeleton is the microfilaments and the microtubules and all of that, as well as attachments to the extracellular matrix, which is the collagen fibers and the elastin fibers. And this one is going to be peripheral, both internal and external. The protein that does enzymatic activity, it could be an entire transmembrane protein. It could be a protein that is spanning the entire membrane, the plasma membrane. But its active portion of it, the part that is doing action, might only be on the inside. Same thing with the signal transduction, cell-to-cell recognition, and all of these are the same way. They can be entirely peripheral or external or internal, or they can span it, but the functional part of it might just be on the outside or inside. Just do kind of an overview of what each one of these things might mean. Transport proteins. In terms of transport proteins, you can have both active transport. There are different types of transport proteins you might have. One is active transport, and the other one is passive. This is passive transport, and this is active transport. In the case of passive transport, it is doing simple diffusion. So things are moving from a high concentration to a low concentration without use of any amount of energy. In the case of so there's no ATP being used in this. It's just moving in terms of the concentration of molecules. In this picture, you will notice that there are more molecules on the outside. The blue molecules, there's more of them on the outside than on the inside. So the molecules are just going to passively move inwards along the concentration gradient till there is the same number of molecules on the inside and outside. So you're going to see till there is no net movement, right? If the concentration gets very high on the inside, then it'll just start moving in the opposite direction. So it's going to move passively back and forth till the net movement is zero. In the case of an active transport almost always goes across the concentration gradient. Passive transport is going along the concentration gradient. That means it is going from a high concentration to a low concentration. In the case of passive transport, it is going from a low concentration to a high concentration. So let's say your cell is trying to expel something out. There is a high concentration of that inside the cell, but then it needs to push it outwards where there is a low concentration of it. So in that case, it is going to use an active transport. An active transport usually is going to require a use of energy. So if you're going across the concentration gradient, you need to add energy to it. Think of it as a pump where you have to like pull it upwards. Whereas if you're going along the concentration gradient, then you just don't have to use energy. A good way to think about it is also if you're driving downhill, you can put your car in neutral and you can just coast. Whereas if you're going uphill, you have to hit the gas. So you have to use more energy there. The next type of reaction is where it does enzymatic action. Enzymatic action, enzymes are biological catalysts. That means they reduce the activation energy for a reaction to take place. They do not take part in the reaction, but they are able to reduce the amount of energy required for the reaction to take place. For any reaction to take place, there has to be two conditions that apply. One is that there has to be enough number of collisions. Two is there has to be collisions in the right orientation. So enzymes usually have an active site. This is the active site where it holds the substrate and allows for reactions to take place. We kind of looked at this a little bit in chapter 5 when we talked about how enzymes are able to break molecules down by using by doing hydrolysis reactions and stuff like that. So that's what an enzyme is. This site is called the active site. The next important function that they do is signal transduction. Let's say there is your body is creating a flight or fight response. Your body has pumped out the hormone epinephrine. Epinephrine now has to binds to the cells in your cardiac center and increases the amount of blood being pumped. Your heart rate increases more. There's the greater cardiac output, and all of this stuff is happening in order to facilitate either you confronting the situation, basically either confronting a threat or taking off. Both of them require a lot of energy. So when that's happening, as the epinephrine molecule goes there and locks into one of the receptor proteins on your cell, and then that signal is sent into the cell to allow the cell to start producing more ATP because you are going to need energy, and that signal is amplified as it goes into the system. So for every molecule of epinephrine that locks in, I'm just throwing out a number, the signal is amplified tenfold, and so that is signal transduction, and this is done by signaling molecules too. The next thing that your cell also does is cell-to-cell recognition. So this is usually done by glycoproteins and stuff like that are sticking on the outside, on the periphery of the protein itself. And for example, your immune system needs to, if you have like a bacterial infection, if you have amoebosis, which is a eukaryotic cell, your immune system has to have a way in which it recognizes cells that belong to you and cells that are foreign to you. And the way it does that is by using the receptors that are on top of your cell that your immune system recognizes. So that is cell-to-cell recognition. For example, if you have an autoimmune disease, that means that your cell, for some reason, your body is not recognizing the self receptors that your cell has. Rheumatoid arthritis is a good example for that, where your joints, your immune system starts attacking the cells in your joints because it doesn't recognize it. So intercellular joining, for example, when we talked about in chapter 6, when there's tight junctions, cells kind of pack the proteins kind of pack onto each other and create like a tight junction. You also need different types of the fibronectins, the integrin proteins that attach the proteins to the extracellular matrix itself. So this is what we were talking about in terms of cell-to-cell recognition. They're usually known as glycoproteins, which is glyco comes from the word carbohydrates, and glycolipids. Glyco comes from the word carbohydrates as well, and lipids is from lipids. Glycoproteins comes from the word glyco, from the carbohydrates, and proteins from proteins, whereas the carbohydrates are covalently bonded. They are sharing electrons, they are not transferring electrons. Both cell-to-cell recognition and all these recognition molecules are usually glycoproteins or glycolipids. The reason you have a carbohydrate there is because you need to have some sort of a distinction, because you just don't want to have just a lipid chain just going off of it, because then it becomes, if you had a lipid, if you wanted to connect this lipid to the hydrophilic So if I wanted to connect this lipid to the hydrophilic side, I would need something that is hydrophilic also to be connected. So between these things, so you have a lipid molecule on one side and the carbohydrate then connects to the hydrophilic side. So this is your hydrophilic head, these are your hydrophobic tails, and then you have a carbohydrate here, which has kind of a nonpolar end on this side, and then you would have the lipid molecule sticking out. Or you would have, this way, you would have multiple carbohydrates sticking out that way. They're usually it'll be like, so the better way to look at it would be, let me draw it out here. You have this way, and then you have a protein in the middle, and in this area where it's interacting, you're going to see a carbohydrate portion of it, and then either a protein or a lipid or whatever sticking out of there. This is another way in which there is transportation takes place. This is the thing that we talked about where we said it goes from the ER to the Golgi apparatus, and then the Golgi apparatus is making is putting these little tags. These tags are made up of glycolipids. So this is the protein, and these are tags. These tags are usually address tags, and this is the vesicle. The vesicle then, based on where this address tag, the one that's sticking out, as you can see right here, it's kind of sticking out, that tells it where it's supposed to go. If there is supposed to go outside the cell, then it just fuses with the plasma membrane. Remember, this one is made up of phospholipid bilayer, the vesicle is made up of phospholipid bilayer, so it fuses in, and then the receptor stays, the glycoprotein stays right there. So when this molecule, let's say there was a bunch of these molecules need to go in, it can form, it goes up there. These molecules will just go up there, connect, like you can see right here, they connect there, and then the vesicle will form on the inside, and then it'll go wherever it's supposed to go. Because all the membranes are made up of the same material, these vesicles can be formed very easily by pinching off one organelle and fusing with another organelle. If they were made up of different materials, it would be a problem, because then ultimately what would happen is either the Golgi apparatus would completely be finished, and then you would have to produce more of its membrane, or the ER would be completely consumed and you would have to produce more of it. Because they are all of the same membrane, there's just pieces of it can be pinched off from the plasma membrane and go and attach to the Golgi apparatus. It can be pinched off from the Golgi and then go and attach to the plasma membrane, from the endoplasmic reticulum to the Golgi, and back and forth it can go, because they are all made up of the same phospholipid bilayer. One of the very important tasks of the plasma membrane is to make sure that it controls what materials goes in and out of the cell, and this is why that the plasma membrane is selectively permeable. Its polarity gives it an ability to be selectively permeable. Molecules that are hydrophobic molecules, the ones that are nonpolar, such as hydrocarbons, whether it is a methane, hydrocarbons. If you remember from chapter 4, I think, hydrocarbons and alkenes, alkanes, alkynes, those kind of things, they can easily pass through the phospholipid bilayer because they are nonpolar, and nonpolar things will allow other nonpolar things to go in. Alkane, ene, and ine. and other lipid molecules can also go in. But polar molecules, one of the easy ways for you guys to tell which molecules are polar and which molecules are non-polar is a very rudimentary way for you to do that is to look for the existence of oxygens and how many oxygens are present. So polar molecules have a much more difficult time going through the membrane because polar materials are dissolved in other polar materials. A large chunk of your plasma membrane is actually nonpolar. One of the ways in which hydrophilic molecules go in through the system is through channel proteins. Channel proteins are these transmembrane proteins that go through the membrane. So this portion of it is going to be hydrophilic. I'm just going to go PHI. The outside is hydrophilic. And then let me change the color. I think it doesn't let me do that. Oh, pen color. Red. This portion of it where it is interacting with the phobic tails is going to be hydrophobic. And that's on the outside. On the inside, the inside of this thing is going to be very much hydrophilic. So the inside is going to be hydrophilic because it is allowing other hydrophilic molecules to go through. So the inside of that molecule is going to be hydrophilic. There are channel proteins like aquaporin, porin meaning a pore, aqua meaning water. So these allow water to go in, and water goes in through a simple diffusion. You are not using energy to send water in. It's going along the concentration gradient. If there's a lot of water on the outside of the cell, then it just diffuses into the cell. If there's a lot of water on the inside, it passively goes outside the cell. Because you know that water is a polar molecule. There are other types of channels called as carrier proteins. These carrier proteins will actually bind to a molecule, change its shape. It changes the shape of the protein itself and then allows that molecule to be shuttled across the membrane. Carrier proteins usually work in terms of they have ports. So as we go along, we'll talk more about ports. The changing of shape is called also called conformational change. So as we go along, you'll see this word come up a lot, where it says a change in conformation. That means it changes the shape of the protein. And proteins are able to have this hydrophilic side, hydrophobic side, and are able to change their shapes based on their interactions and stuff like that because they have amino acids that are polar. They have amino acids that are non-polar. That they also, when they form their secondary structures, tertiary structures, and all of these structures, there's a ton of hydrogen bonds involved in them. So they're able to make these conformational changes based on the hydrogen bonds connected to them. Transport proteins are very specific. A transport protein that transports, let's say, a sucrose molecule is going to only transport sucrose molecule, or at least it's going to have a port system with which the sucrose molecule goes in one direction and maybe ATP or something goes in the other direction, but they are going to be very specific to the protein, to the substrate that they are moving across. One of the things before we go into how transportation takes place across the cell, let's just do a basic review of the chemistry here. Diffusion is when molecules are going from a high concentration to low concentration. They are traveling along the concentration gradient. Diffusion is a passive transport. You don't have to do anything. You do not have to provide energy to it. It just gases as a nature tend to occupy the space that it is provided. That is why gases don't have a definite shape or a definite volume. So these molecules will randomly diffuse from a high concentration to a low concentration, and they will keep going till the net movement is zero. So if I have a semi-permeable membrane and I have 10 moles of molecules here, and I have 8 moles of molecule here, then it's going to keep going in that direction. There'll be a high movement in that direction. And once that and that both become 9, there is still going to be movement in that direction as well as this direction. But the net movement here is going to be 0. That is what a dynamic equilibrium is. So this is what I mean by net diffusion. So you have a semi-permeable membrane, and this is all based on a solubility. So if you remember, solubility is a solution has a solute and a solvent. So this is based on concentration, right? Moles over liters. So if I had a solution, let's say I had this, let's take this one. Water is going to, there's more solute on this side than there is on this side. So the solute molecules are going to move in this direction, and water molecules are going to move in that direction. And that's what this thing is going to happen back and forth till there is an equal number. It's going to go back and forth till there is equal number of solute molecules on this side, solute molecules on that side, solvent molecules on this side, and solvent molecules on that side. That doesn't mean that there is going to be no movement. It just means that the number of H2 molecules moving from that side to this side and H2 molecules moving from this end to that end is equal. And the number of these solute molecules moving from that end to that end and that end to that end is also equal. Both of these are going to be equal. And that's what a dynamic equilibrium is. We talked about both active and passive transport. Anytime something is going along the concentration gradient and is using diffusion, it's going to be a passive transport. And you might have heard of this word called osmosis. Osmosis is diffusion of water. So when molecules other than water are moving from a high concentration to low concentration, we just call it diffusion. If it's water molecules moving from a high concentration to a low concentration, it's called diffusion. It's called osmosis, sorry. Osmosis is diffusion of water. Water is going to diffuse again along the concentration gradient. I'm going to stop right here because after this is isotonic, hypertonic, hypotonic, and it can get a little confusing. So I'll do that in class.